Yeah, right. Today we have Francesca. I think she's one of the rising stars of Europe, and she has worked in a lot of places with a lot of big names, and her publication speaks a lot for her. So at the moment I can only say that she's going to rise high and high, and we are going to know about light sheet as well as super S, because she's expert on both. So let me, I will not speak too much. Already your bio is there with us, so we can see it through. So Francesca, over to you. I mean, please go ahead. And yeah, we are looking forward to listen to you. Thank you, Francesca. Bye.
So
yeah, please go.
Thank you, Professor thank you, Professor Mondal, for the nice introduction. And thank you, Parth. I will start sharing the screen so you can actually see. Let's share this one. So can you see my shared screen?
Yes,
hopefully.
Yes.
Okay, perfect. So thank you very much for the nice introduction, and I'm really happy to have this talk to this nice workshop. And I will try to focus basically on, as Professor Mondal mentioned, on super resolution techniques. But I will try to look at it on the other side. Like, okay, I'm not focusing on super resolution itself. I'm focusing on how the marriage between light sheet microscopy and super resolution can gain advantages in bioimaging. So the main challenges are the fact that we have super resolution techniques, we can use them, but then there are still a lot of challenges. How to improve these techniques, how to go, for example, towards tissue imaging and three-dimensional imaging, how to match the sample requirements, because if we want to address biological problems and applications, we need to match the requirements of the sample. So having the technique itself is nice, but is not enough. The technique needs to go close the biology to match the requirements from the sample. The other side is how we can use these techniques not only as imaging techniques, but how we can use them as a real quantitative tool. So these are the main challenges, and I will try to look at them and make a panoramic on how we can solve these challenges. So first we have to consider that, I will try to remove this guy here. Let me see. Okay. So basically, when we are working with super resolution, we have to consider that in the first and nowadays, the technique is well established, especially at the whole cell level. So if you want to work with cell cultures, everything is fine. If you want to use it for imaging, the technique is already established. But what about if we want to push this aspect further? So the basic idea is that we are dealing with scattering samples. So we have to consider that if we want to move to tissues or three-dimensional samples in general, we are dealing with scattering samples. So we have three ways of approaching and three ways to improve the performances. So we can work on the optical approaches, so changing the imaging system. We can work on the dyes we are using, so changing the labeling procedure, going to use new molecules or new dyes with better performances, or we can have new, for example, algorithms for super resolution. Then we will see which kind of super resolution in details, but for the moment, this is. So first I want to focus on how we can do, what we can do from the optical approaches side. So one aspect is we can think about combining light sheet fluorescence microscopy with super S. So the basic idea of light sheet fluorescence microscopy, as you know, at the third days of the workshop, you saw it in details before, The idea is to have this planar illumination and confining the illumination only on the focus, on the focal plane. This brings a lot of advantages, like optical sectioning, an intrinsic optical sectioning capability, the possibility to have fast imaging, and a good high signal to noise ratio. And also we can reduce the photo damage on the sample. So basically, this is a system. Here you can see an optical scheme taken from the literature from the Stelzer lab, for example. Here you can see one of the simplest implementations where we have the illumination side and the detection side in a selective plane illumination microscopy scheme. But there are different implementations. For example, there is also the digital laser scan microscopy. These are all examples taken from the literature. Here you can see this paper was published on Science, where a scanning version, so a beam was scanned to create the light sheet, was used instead of the static light sheet created by the cylindrical lens. So these are two different implementations of light sheet microscopy. Both of them have similar advantages, but also they have also some key points. For example, the selective plane illumination microscopy is more easy for the implementation side. And on the other side, the digital laser scanning microscopy is, for example, suitable also for beam shaping and beam shaping implementations. So depending upon what you want to do, you can choose one of the two architecture, but both of them has optical sectioning capabilities and high speed, for example, possibilities. So just this is just to mention that depending on which is your sample, what you want to image, you need to consider or adjust, I would say, the optical scheme to have the correct field of view. So this means there is a relation between the field of view you can reach and the thickness of the light sheet. So then you have to choose to make a balance. Okay, I want to image large samples, then I have to have a big field of view, but probably we will have also a larger thickness of the light sheet, so lower axial resolution. So this is just to give you the idea that these parameters that are defined by numerical aperture of the illumination are quite important, and you have to think about which is the sample you want to image and then go for the best axial super resolution, thinking about also the field of view you want to reach. So one of the main advantages of light sheet microscopy is also the fact that you are confining the light. And confining the light compared to confocal and conventional microscopy means that you are also reducing the photobleaching effects within the sample. So this is why this technique is basically used, for example, for developmental biology, where you have live samples, you want to image them, large samples for a long time. But this brings also a good axial resolution. Here you can see the imaging of fluorescent beads as a test sample. For example, this is also taken from the literature to give you an idea of the advantage you can reach. And here you can see the axial resolution when you're imaging with wide field, where there's no axial resolution, or you have the lens resolution, compared to the light sheet microscopy architecture, where you have a more isotropic resolution. This is why also, this is a simulation where you can see the point spread function of the system, that you can compute it by thinking about the point spread function or the illumination intensity distribution of the light sheet multiplied for the intensity that you can reach from the detection point spread function, And this gives you the overall point spread function of the system that is basically more isotropic compared to the wide field microscopy scheme. And this brings clearly an advantage in terms of axial resolution and contrast. Here you can see in a real sample. This is also a paper from 2011 taken from the literature. You can find all these things in the papers published. But this gives you the feeling that this technique is quite helpful for imaging. You can do multi-view, for example. So the fact that you are sectioning with the light sheet gives you the possibility to make single views, so optical sectioning by, or plane that are parallel one to each other, or you can also image from different angles. When you are imaging from different angles, you can push the resolution and you can gain information and combine them all together. And this brings, like what you can see in this example, brings the resolution is even better, and the axial isotropic resolution that you can gain brings a good contrast in the three-dimensional image. Here you can see a nice example where you can follow a wild-type zebrafish embryo that has been injected with GFP to the H2B histone at the cell stage level. So you can follow the development of the whole embryo over the time for long-term imaging. And this gives you the powerfulness of this technique. But the basic idea is to, having this sectioning by the plane. And if you think about the wide field, in the wide field, we saw that you are illuminating the whole sample. So light is traveling through the sample, and you have, the problem of the resolution, the axial resolution, but also the fact that your sample is illuminated. So this is the main issue. So you can move when you want to do super resolution. For example, one of the first approaches was to move to inclined illumination. So trying to incline the beam as much as you can to have an axial confinement of the illumination, so a better contrast. But if we want to bring to the extreme this aspect, you can think about moving to selective plane illumination. That is, is inclined illumination brought to the limit of 90 degrees. So how we can increase the resolution? Because now we saw that light sheet microscopy is a really nice tool. We can use it. I showed you from the literature some examples. You can use it to image large samples, follow dynamics in embryos with high contrast and good optical sectioning. But what about if we want to push and increase the resolution? We can think about using super resolution, and in particular, single molecule localization. So the basic idea of super resolution, I will be fast, but I will give you just the idea of the technique. The fact is that you are going to image within the sample fluorescent probes that are really small. So they are on the range of 2 nanometers, 3 nanometers, usually, even smaller for synthetic dyes. So the point is that we are going to image our probe that is really small with a microscope, and the microscope has a finite point spread function. So the resolution is limited. So we will see a spot that is limited by diffraction. This means that we cannot resolve objects within our samples that are closer than the diffraction limit. So the distance that is given by the Abbe's law, as you can see here. And if two objects are closer than this distance, that depends on the wavelength and the numerical aperture of the lens you are using. This means you cannot resolve the two objects if they are closer than this distance. But what about if we can switch on and off when we want our fluorescent probes. So if we can identify two states, the dark state and the bright state of our fluorophore, then we can switch from one to the other in a controlled way, usually controlled by light. We can play a game. So the idea is that we are still using conventional microscopy. So we are still using a microscope, a wide field microscope, for example. So we have a point spread function of the sample of the lens and the system, and we are going to image an unknown sample. But what about if we don't image all the molecules together, but we can separate them in time? So the idea is that we switch on only few molecules. So here you can see few molecules that are far one from each other. And we can, since they are separated, we can identify by processing, or we can compute the center of each molecule, so the centroid of each spot. We can map the center, and then we can switch the molecule off, because we saw that we can move from the on to the off state in a controlled way. So we switch off the molecules and we turn on other molecules, and then we repeat on, off, localize, on, off, and localize until we have an information, a final image, a reconstructed image, because this is only a trick. We are not really changing the microscope. We are playing with the molecules to play this on-off game to reach an information that is definitely far below the resolution limit. So here you can see how does it look an experiment, and the idea is that when you image each of these single molecules within your field of view, you can localize them. So the localization process is given by the fact that you can have a precision in saying where is the center of each molecule. And this precision depends on the parameters given by the imaging system, by the microscope. So that the point spread function of the microscope, for example, plays a role, but in particular on the molecule, because it depends on the number of photons that you can collect. So as many photons you can collect, better will be your localization precision. So here you can see an example in a cell. Here you have cells where microtubules has been labeled with Alexa 647, for example, and you can switch on and off the molecules, record enough frames, and in the end, you can have a map of super resolved map of the molecules you localize. And this is how it seems a single molecule localization image. You can see that it's color coded from green to red because it's also a 3D technique. So we can also assign, using astigmatism, for example, the axial position with high resolution. So basically, we have a 3D technique to localize molecules and provide a super resolved image. Which are the parameters that can be important in the localization process, as I told you? One is the number of photons collected. So as many photons we can collect, better will be our reconstructed image because our localization precision is improved. On the other side, if you are looking to thick samples or tissues, as we were mentioning in the first, if this is the aim, so going towards tissues, there is the factor B, that is the background noise, that is increasing. So if the more is complicated your sample, higher will be your background noise and worse will be your localization precision. So here is just to say how you can define the localization precision. And the point is that the molecule position is actually computed as the mean of the position of each individual photon detected. Okay, and the error on this measure is the standard statistical error. That's why our localization precision depends on the inverse of the number of photons collected. And here you can see there is an additional term that also takes into account the background noise. Here is just to give you an idea or on which is the range where you have to be placed to perform single molecule detection, because it's not so easy. You don't go with your sample, you put it under the microscope, and then you see molecules popping out. The point is that finding the right laser intensity. So the laser intensity is quite important, and in relation to the fluorophore you are using. So there will be fluorophores that will be suitable for single molecule localization and fluorophores that are not so optimal for the switching from the on to the off state. But as you can see the fluorescence lifetime and the triplet lifetime. If you think about the Jablonski diagram, your molecules can move to the triplet state. It's a non-allowed transition, but it can happen if you increase the laser intensity. So the region where you can perform single molecule detection is the region comprised between the fluorophore lifetime and the triplet lifetime. And this is actually corresponding to high laser intensities. Okay, for example, 1 kilowatt per centimeter square, usually. This is the range. So we can use this triplet state transition to make the molecules dark, because the triplet is a dark state. Or we can use, for example, other processes like photoactivation or photoconversion. So here there are examples of photoactivatable fluorescent proteins, for example, that can be used. So this protein has no absorption spectrum before being illuminated with 405 with UV laser usually, and they become fluorescent after the radiation with the photoactivation laser. So this means the molecule was not a fluorescent molecule before radiation, before photoactivation, and it becomes a fluorescent molecule. This is the way you can play the on-off game. On the other side, you can also use photoconversion. So for example, you can change the color of your protein. So you still irradiate the sample with the UV light, usually 405, and your molecule, for example, usually the transition is from green to red, but can also occur on other spectral regions. But the basic idea is that our molecule has an absorption and an emission spectrum before UV radiation, but this absorption and this emission spectrum change after radiation. So we can select with filters another spectral region, and we will see the same effect. We have on and off states that we can use. So this is the idea of single molecule localization microscopy and super resolution. But let's go back to the starting of the talk. We saw how nice is light sheet microscopy and the basics and which is the advantages we can gain. But the question is how we can use light sheet microscopy to improve the resolution. So how we can combine it with super resolution and try to make super resolution of larger tissues, not only cells or thin samples. So the basic idea is trying to go to tissue imaging, basically. So what usually people want is to image, the dream is to image a tissue directly. So let's go with, step by step. So we have to consider that we have a scattering sample. So we need to think about how to reduce the scattering and the absorption in order to improve the imaging depth capabilities. Because if we try with the normal single molecule localization architecture, then if you want to move inside the sample and you want to go deeper, then you will lose the capability to detect single molecules, because the light is not efficiently reaching your fluorophores, And also the emitted photons will get lost while going back to the lens for the detection. On the other side is the question is, which is the sample you want to image, because tissues means, for example, brain slices or retinas or whatever, it has a morphology, okay? And light sheet microscopy, has some requirements from the sample handling. So we need also to think about how to optimally hold the sample within the light sheet microscope. As I was mentioning before, for super resolution, the main advantage of light sheet microscopy is the confinement. But the point of this confinement is that we are not exploiting molecules that are within the sample. Because when we use wide field microscopy, we are photoactivating, photoconverting, or bringing to the triplet, whatever you want to, which kind of technique you want to use. But the basic idea is that the conversion and the illumination occurs within the whole sample along the axial direction. So this means molecules, when you will move to another position within the sample, another plane, another thickness, the molecules will already be, they went for multiple cyclings, they already have been photoactivated, so you will not have a good performance. So the basic idea is that using light sheet microscopy, we can preserve the molecules that are above and below the focal plane. So we can then move and we can still image them with high performances. So this was one of the first light sheet architecture I was building in the Alberto's lab, in Diaspro's lab. So he already mentioned you some key aspects of how we can combine light sheet and single molecule localization microscopy. We can now go more in details and see the details of how you can reach this combination. So here you can see the illumination unit is, it has two possibilities. It can work with visible light, but it can also work with infrared light for two-photon excitation. Then we will see how and why. Then we have a cylindrical lens, because this is a selective plane illumination microscopy. Similar things can be done also with the scanning version, but now let's see the simpler one with the cylindrical lens. And then we have a detection path. The detection path is quite similar to a wide field path, but it has an additional magnification that is required for detection of single molecules. So you need the right magnification for imaging single molecules. So here you can see the first optical scheme we built. And the first step was checking that we can have photoactivation in the light sheet regime. So the basic idea is that we can image cells. This is just a test. We were expressing a photoactivatable protein PM cherry within with the histone H2B. And we can see when we switch on the activation light, so the UV light, the signal is increased. So we are really photoactivating using the light sheet within the sample. So we started with thin samples as a test. These are polyelectrolyte nanocapsules. So this is a thin, small, and transparent sample. So we are still not going to tissues, but we are preparing the light sheet architecture for single molecule detection, thinking about tissues. Here you can see that you can compare the conventional selective plane illumination microscopy image with the super resolution image implemented in a light sheet configuration. We can also do 3D super resolution using an astigmatism approach. So we are inserting a cylindrical lens also in the detection path, so we can have a distortion, and the distortion depends on the position along the axial direction. So from the electricity of the spot we can image, we can also assign the axial position with high precision. So now the idea was to go to tissues. So let's think about going to thicker samples. So first, we can think about spheroids, for example. Here we have a cell spheroid, a human mammary cell spheroid. We need to think about when we are imaging such a kind of sample that is like hundreds of microns thick. We have to deal with the fact that light is traveling both along the illumination path and the detection path. And in both the cases, there is effects induced by scattering and absorption. Here you can see the final image. So you can see how we can improve, because we have a super resolution image of the cell spheroid within the light sheet architecture compared to the conventional selective plane illumination. Here you can see we have roughly, I would say, 28 nanometer of localization precision. So let's be careful. Localization precision doesn't mean resolution itself, but it gives you the idea of how good is your ability to localize, and also this means also which is your resolution in the final image. So the imaging depth you can reach, for example, this image was 100 microns deep within the mammary cell spheroid. So basically, here we can, this 28 nanometer of localization precision I mentioned is a theoretical one. So is the localization precision that we can compute considering the experimental parameters. So there are analytical models like this Mortensen model, where the localization precision can be calculated, and it depends on the point spread function of the system, the pixelization of the system, the number of photons collected, as I was mentioning, but also the background noise, as we were looking before. But this is not the real localization precision. This is not the effective one. The effective one can be calculated, the real localization precision. Needs to think about also about the taking into account the instabilities of the system. So the fact that you have a larger sample, the fact that there are different sources of instabilities like vibrations or any other problem induced by the thickness of the sample and the system. So basically we can compute this effective localization precision by adding the terms like the theoretical localization precision and the sigma instabilities, taking into account about all these factors. And then we can go to the real sample, so we can go inside the mammary cell spheroid, and we can try to measure the effective localization precision. So if we find single molecules and we make a repeated localization of molecules within the mammary cell spheroid, so deep into the sample, we can have a distribution in the radial and in the axial direction, and we can compute the accuracy. So our precision, for example, the accuracy as a full width half maximum. So the one along the radial direction is around 63 nanometers, and the one along the axial direction is 141 nanometers. So this means a factor of 2 compared to the theoretical localization precision I was mentioning. So this is, there is a decreasing in the localization precision due to the thickness of the sample, but this is still a good value, because thinking about having a technique that goes into tissues and can reach 60 nanometers of localization precision or 141 in the axial direction is really a good value. Still thinking about the fact that it is worse than the theoretical one. But then we need to optimize the system. We need to think about, I was mentioning, the fact that light is traveling through the mammary cell spheroid or the tissue you are interested in, along the illumination side and also the detection side. So we have distortions we need to take into account, or we need to verify that there is no effect. So first, we can test what happens along the detection side. So we can move within our mammary cell spheroid, and we can check the performance in localizing molecules. For example, in this case, we are 10 microns within the sample. Here we are 40 microns, 70 microns, and 100 microns within the sample. So we can still localize molecules along the whole mammary cell spheroid, because one option was, if we move deeper into the sample, we lose photons and we end up at the, on the other side of the mammary cell spheroid, we cannot see, for example, single molecules, but this is not the case. What we need to test now is if we can still collect the same amount of photons within the mammary cell spheroids at different depths. Why this is important? Because if we are losing photons going deeper, then also our localization precision will be worse and worse when we are going deeper. So this means we could have a localization precision that is depth dependent. But this is not the case, because if we look at the number of photons per molecule at the different depths, so 10, 40, 70, 100 microns, we can see that they are still in the same range. So there is no loss within the mammary cell spheroid, and we can consider that we have a localization precision that is depth independent. The other option is we need to check that the light sheet that is traveling along the illumination side, the illumination is not getting degraded while it goes into the sample. So it goes into the sample, and your light sheet could be, if there is a lot of scattering and absorption, brighter, so the illumination can be more efficient in the first part of the light sheet, and then it goes decreasing while entering the sample. So also in this case, this is not the case, because if we look what happens 50 microns in the sample, 100 microns or 200 microns within the mammary cell spheroid, we can still see kind of flat illumination, so uniform illumination. The last check we did is to check that the point spread function, so we can image bits in a scattering sample and in a non-scattering sample, and we can see that there are no aberration and distortion induced by the scattering effect. And then we move to the real sample. Here you can see, for example, in the mammary cell spheroid, you have connexin 43, that is a protein that was labeled with PM cherry as a fusion protein. And you can see, different planes, 1 micron by 1 micron, and you can also see the color-coded map with all the information overlapped. So we saw how we can increase the resolution, how we can combine light sheet microscopy and single molecule localization microscopy to improve the resolution. But that is a step further we can do, because what about, okay, we like the mammary cell spheroid, but what about if we would like to push this imaging technique to larger samples? So, for example, tissues or less transparent tissues, for example, less transparent than the mammary cell spheroid. So what we can do is to think about using two-photon excitation. So the basic idea of using two-photon excitation is that we can use wavelengths that are higher, so reducing in this way the scattering. Here you can see an example. This is a brain light sheet imaged by two-photon light sheet microscopy. You saw when I showed you the optical scheme that it was also equipped with a mod-lock laser, an infrared laser for two-photon excitation. So here we can do normal imaging with two-photon implemented in a light sheet architecture. Here you can see the infrared lasers that has been coupled to the system. And we can also see that the distortions, because if we look at the profile of the light sheet in single photon excitation compared to two photon excitation, we can see, we can check which are the distortion in the intensity profile. And also we can calculate the contrast. So if we look at beads, for example, we can see that the contrast is more robust if we use two photon excitation. So here you can see that when you're using transparent, when you're imaging transparent samples, the contrast is the same if you're using single photon excitation or two photon excitation. So this is not helpful for transparent or thin samples. But if you want to go to a thick sample, so if you want to go in depth, so deep into the sample, and the sample is a scattering sample, when you calculate the contrast, you can see that the contrast is performing better, is higher, if you use two photon excitation compared to single photon excitation. So here you can directly see the contrast decreasing. In the first line, you can see single photon excitation, in the second one, two photon excitation, and you can see that there is an improvement in the contrast, increasing the scattering coefficient of the sample. So here we were using a phantom sample mimicking different scattering coefficient values. And you can see that if we increase the scattering coefficient, single photon excitation performs worse, is not so optimal compared to two photon excitation that is more robust and has a better contrast also for high scattering values. Here, as an example, you can see the reconstruction of a 3D image of a mammary cell spheroid. And we can also characterize the light sheet thickness. Then we will see why I'm characterizing the light sheet thickness, but the point is that our light sheet, if we are using single photon excitation in scattering sample, when we are going deeper, what's happening is that the light sheet, due to scattering, is getting larger and larger. So as much deep you are traveling into the sample, the larger the light sheet will be. So this is not happening for two photon excitation, because when we're using two photon excitation, there is no this distortion, and the light sheet thickness is the same going deep into the sample. What does it mean? This means that in thinking about single molecule localization, if we are working with the non-scattering sample, fine, we are illuminating molecules in the focal plane. But if we are moving to scattering samples, what we are doing is going into tissues or thick samples, going deeper, will produce a larger region of molecules that has been illuminated, excited. So this means we are not confining properly our photoactivation, for example. But if we are using two photon excitation, we are actually confining always in the very same way, even if we are moving to scattering sample and we are going deep into the sample. So this means we are still preserving the possibility to perform, photoactivation of a plane of molecules within a plane and then move to another plane, preserving the molecules that are above and below the focal plane. So here you can see an example. Here there is some an image of H2A, phospho H2AX labeled with the photoactivatable dye. In this case, we were using two photon photoactivation. So the photoactivation was performed using 760 nanometers instead the visible light. Readout is still in the visible range, but the photoactivation was confined into the volume using two photon excitation. Last aspect I wanted to mention is the fact that we need to match the sample requirements. If we want to address biological questions, we need to go closer to the sample requirements. One of the aspects is the sample handling. So we have different architectures for light sheet illumination. For example, we can use the conventional light sheet architecture with the light sheet and the sample hold in the water chamber, or we can perform an inverted architecture. So we can build an inverted SPIM. Optically speaking, is quite the same. The point is that the illumination and the detection object comes from the above, and this means you can also work with the Petri dish, for example, if you have your sample, or with a slide. Why this is helpful, for example, here you can see the architecture, you have the illumination from the right and the detection from the left side. The point is that you can, for example, image properly tissues like the retina. So if we think about the mouse retina, we have different layers like the ganglion cell layers, the amacrine layers, and the photoreceptor layers. And the key point is that we need to use the inverted configuration, because if we try to hold the retina within the capillary or within the water chamber, we will lose morphology. So it will be hard to identify the three layers, and we will lose completely the shape and the morphology of the sample. In this case, we can still maintain the morphology of the sample, and we can image and we can look at the three layers, like the ganglion cell layer, and then the amacrine cell and the photoreceptors. So this is important. It's a small change from the optical point of view, having the inverted configuration, but really opens the possibility to image a wide range of samples. Similar considerations can be done for the brain slices, because also a brain slice cannot be easily held within agarose gel or in the water chamber. And we still need, so here we can see for sorry, the screen, the share screen disappeared, or you can still see my share screen. Let's see, share again. So let's go back to where we arrived. Here we are. So the basic idea is that we can have a higher penetration depth using two-photon excitation, but we can bring it also to brain slices, because we are using this inverted configuration. So only the combination of different aspects, like using multiphoton, using the inverted configuration, can bring to a successful imaging of tissues. Here, as I was mentioning before, you can see the image of the brain slice. And now it should be more clear why we need the inverted configuration. I wanted to mention, this has been taken from the literature, also another option that can, bring the technique closer to the biological question. And this is the single objective SPIM. In this case, some micro mirrors will bring the light on the focus of the detection lens. This is another option. So we have the conventional one, for some samples is fine, the selective plane illumination architecture. For other samples, an inverted configuration is required. And also this single objective configuration can be used. I wanted to tell you only a an outlook. Another aspect that can be improved, because now we saw how to match light sheet and super resolution, but another aspect is how to bring the quantitative capabilities of super resolution, because super resolution can be used, especially single molecule localization microscopy, as a quantitative tool, because you are looking single molecule, so you can be really quantitative. And you can also implement this quantitative technique combined with light sheet microscopy into tissues. So the basic idea is that we can think about going and quantify single proteins and directly within the tissue. And this is really important and helpful. I just wanted to give you the take-home message and just make a brief recap of what we talk about. So we spoke in detail how the marriage between light sheet fluorescence microscopy and super resolution, in particular single molecule localization microscopy, can help and can bring us closer to a real tissue super resolution technique. So how we can really think about to do a successful super resolution imaging in tissues. We also saw how two photon excitation can participate and bring improvements in this scenario. And also how is important to think about the matching of the biological sample requirements and the optical technique, because if we think about all these aspects, we can really reach high quality imaging in tissues. So I would like to thank you for your attention, and I will be happy to take questions.
Yeah, thank you, Fra. It was really a absorbing talk, and your contribution in unifying light sheet microscopy with single molecule is specifically a great step forward. So we'll just go ahead and see if some of our some of them have some questions. So Jigme, can we have some questions if some of them want to ask some questions?
Yes, sir. Like offline audience, do you have any questions?
Yeah, please.
It seems no one has, sir.
Can you? Oh, okay.
One question, sir. One student has.
Yes, go ahead. Please go ahead with the question.
One second, sir.
Hello, am I audible? Hello.
Hi, yes, I can hear you.
Yeah, I want to know that what is, with the help of super resolution microscopy, what is the maximum resolution we get?
Okay, it depends on which kind of sample you want to image, I would say, but usually tens of nanometers, so radial resolution. So then it depends on the thickness of your, as we saw, on the thickness of your sample. So if you're looking to a cell, you can reach 20 nanometers. If you are looking to a tissue, probably this will be more 60 nanometers or 70 nanometers, or even worse if it's a non-transparent sample. So this is the range, anyway, on tens of nanometers.
Ma'am, I have a question.
Yeah.
consider two cases. One is normal excitation using visible light. Another one is IR, like 2-photon excitation using infrared light. What is the photo bleaching in these two cases? Is it remain same or is it different?
Well, actually, it's the same for the readout, because you are photoactivating with a laser in the UV, and then you are imaging and collecting the single molecules with visible light. So in this case, we were changing for 2-photon, we were changing only the photoactivation. So I'm expecting in that case that we are preserving molecules from photoactivation above and below the focal plane. So we are preserving that part of photobleaching, but we are still imaging with the visible light. So for the readout, the effect on the photobleaching is exactly the same. So we are reducing only one part of photobleaching, that is the one due to the UV light that is usually used for photoactivation. This in the super resolution. In the imaging, where like I show you the brain slice, for example, this was really reduced the photobleaching because we were using 2-photon excitation also for the imaging. But for super resolution, we can reduce only one part because it's really hard to collect single molecules using 2-photon excitation. So we can use 2-photon excitation basically for the photoactivation, but not for the readout.
Okay.
Any more questions?
Anyone has any more question? No, sir.
Okay. If so, let us thank Francesca for the nice talk and all the advances that she has talked about.
Really, thank you for inviting
Thanks a lot,
me.
Francesca. Yes, we are really happy to have, and to be able to listen to you, and the amount of work that you have done is really commendable. Yeah. So yeah, thanks a lot. Let us say goodbye. I think we'll see you again. Yeah. Thanks. Bye.
Bye. Bye.
Thank you. Bye-bye. We'll end the meeting for all.